So first let's start with this diagram. এই ডায়াগ্রামটা দেখে কি মনে হচ্ছে? What? Okay, so this photograph was taken around 1600, mid 1600 এরকম একটা সময়, and this was the first of its kind. এখন তোমরা যারা সেল বলতেছো, ওই সময় সেলের কনসেপ্টটা তখন এস্টাবলিশ হয়নি। So after the scientists who made this diagram observed this, তার কাছে মনে হলো যে these are just some regularly arranged rectangular pattern. And the first thing that came to his mind for some odd reason is the word cell. Now, remember one thing, এখনো পর্যন্ত কিন্তু সেল কনসেপ্ট এস্টাবলিশ হয়নি। সেল যে কি জিনিস এটা কিন্তু আমরা যেটা সেল বুঝি সে কিন্তু এটা সেল কখনো বুঝতো না। So তার কাছে সেল কনসেপ্ট আসলে কি ছিল? Where did he come up with this name cell? No. Cell কি? What are the definitions of cells? What are the other meanings of cell? It can be cell phone, but then 1600 is cell phone ছিল না, so definitely not that. Does it make sense? Prison cell, right? So for somewhat surprising reason, the first thing that came to his mind was prison cell. We can take a share. এটা নাম দিল কি? Cell. And this wasn't the first case. সে তার ইনস্ট্রুমেন্ট দিয়ে যত কিছু অবজার্ভ করলো, দেখলো যে সবারই এরকম রেগুলারলি অ্যারেঞ্জড প্যাটার্ন দিয়ে আসলে টোটাল অর্গানিজমটা বানানো। সো ফলোড বাই দিস, আলটিমেটলি এটা নিয়ে আরো রিসার্চ হওয়ার পরে দে কেম আপ উইথ দ্য কনক্লুশন যে all organisms are made of unit called cells. Okay. Now এটা কিন্তু কোনো ফটোগ্রাফ না, because mind you back then camera wasn't available. So this was a hand-drawn picture. And the man who drew this picture, or the man who made this observation, was this guy, Sir Robert Hooke. Basically, probably lecha, because I'm not sure if this is the actual picture of Robert Hooke. Interesting thing is that no known portrait of Robert Hooke exists today. দুইটা কারণ। এক নম্বর হচ্ছে রবার্ট হুক, he wasn't born in an aristocratic family like other scientists. He grew up in a very humble beginning. খুবই পুয়ার ফ্যামিলি থেকে আসছে। সো ওই সময় তার সবার ঘরে ঘরে ক্যামেরা ছিল না। In order to make a portrait, you have to hire someone, right? Painter, সে আসবে. It would take months to draw a portrait. So সে এটা অ্যাফোর্ড করতে পারতো না। But then eventually he got famous and he did have some portraits made. But interesting thing is this: Newton, this is Sir Isaac Newton, may have got rid of its president of Royal Society to suppress evidence of Hooke's competing claims over his law of universal gravitational field. Basically what happened is that Robert Hooke, সে শুধু বায়োলজি নিয়ে কাজ করছে তা না, he had also contribution in the field of physics. And Robert Hooke was an exceptionally brilliant man, but his behavior was extremely rude. So he got into argument with other scientists, and one contemporary scientist was Newton. Newton's সাথে he got into some argument, and he managed to piss off Newton so much that after the death of Hooke, Newton became the president of Royal Society, and one day Newton allegedly decided Royal Society তে প্রেসিডেন্টদের তো সবার একটা করে ছবি আছে, রবার্ট হুকের যে ছবিটা ছিল সেটাকে ওটা ফেলে দিয়েছে অথবা বার্ন করে ফেলছে, something happened. So that was the last remaining portrait of Robert Hooke. So you can understand how rude this person was. Pissing off Newton is like pissing off Gandhi, right? It's like Mahatma Gandhi giving a middle finger. So that's what happened over here. Okay, anyways, about Robert Hooke. So in your book, you will see it is said in the page, I think second paragraph, it's written that Robert Hooke was one of the first person to discover or make a compound microscope. And what was the time? I think it was 1650s or something like that. So not only 16 year mother, interesting thing is that he was also one of the first person to discover, তাই না? এখন কথা হচ্ছে যে the British people have written this book, so they are going to give their version. But the truth is, 4,000 years back, Greek also came up with the idea of compound microscope. Egyptians, they also had their own version of microscope. In Iraq, They had their own version of microscope in AD 1000 or something. There was a Chinese version of a microscope during the, I think, Yuan dynasty or something like that. India had their own version of microscope. And this guy came in 1600s and they are claiming that he's one of the first makers of microscope. But we have to accept it. It's called a British curriculum, so we have to say yes to whatever they say. Anyways, moving on. So this is the device that Hooke invented. So his early model of compound microscope. And following the invention of microscope, he used this device to observe or magnify things. And what he saw, he drew up a sketch of them and published this book. This is a very famous book. It's called Micrographia. And these are some of the pictures that has been collected from this book. It might not seem very amazing at this point, standing in 2022. But remember, this was published in the late 1600s, right? So back then, this was a staggering thing. Right? People were really amazed because they had never seen something like this. They have all seen ants and fleas, but at detail, at zoom kore this is probably fungus. So this is one of the first detailed picture of fungus. Before this, people didn't have much idea how fungus looks in detail. And this is even more interesting. You see, back in this time, like I said, cell had not yet been discovered, right? They didn't have any idea about microorganism. So to me, if you went to a pond, you scoop up a glass of water, if it looked clean, it meant that the water was clean. However, after this discovery of microscope, when Robert Hooke took out a scoop of that clear water, and took a drop of that water, analyzed it under microscope, he saw that apparently clean water contained all of this. They had no concept of filtering the water. They didn't boil the water because they thought if we can see anything, there isn't anything in the water, right? But that wasn't true. Water was steaming with microorganisms, and subsequently this led to other discoveries which eventually gave rise to the germ theory, and people became more careful. They started to drink filtered water or boiled water and everything. And another thing is that Robert Hooke was one of the first person to observe all these things, right? And these are not stationary things. These are like little animals swimming in the water. So instead of calling them microorganisms like we call them today, he came up with an interesting term called animalcules. I hope I'm pronouncing it right.
Animalcules.
Yeah, something like that. Animalcules or animalcules. Back in his time, these microorganisms were called animalcules. Okay, now standing in 2020, we have much more refined versions of microscope. Now, you have to understand, the concept of magnification existed several thousand years back. What we're talking today is about compound microscope. The basic version of compound microscope consists of two lenses. So the image focused by the first lens is then projected onto the second lens, and this gives a much more magnified image of the actual object. This is a simplified version of the microscope, and this is the modern light or optical microscope. So this is the common version that you're going to find in school, universities and colleges, etc. So here is a level diagram of the light microscope or optical microscope. We will study this in details when we do the practical segment. For now, we need to just know a few things. The specimen that we are going to observe will be placed over here on a glass slide. Okay, so this is called a stage. So the specimen is put on the stage, and like we mentioned, at a microscope, how many lenses are there? At least two, right? So the first lens is the object lens, which is placed over here. Objective lens, sorry. And the other one is the eyepiece lens, which is located over here. So the specimen is here. It is first magnified by this one, then magnified by this one, and then it goes into your eye. And to make the things more visible, we also need to have a light source underneath. Clear? Mostly is available. Oh, sorry. This is nose of the microscope, not your nose. You can actually rotate it, and there are many different types of lens. Some of them have 10 times magnification capacity, like 50 times magnification, 100 times, and so on. Now, the modern versions of microscopes that we have, they have a built-in light source. But the older versions used to have a mirror instead of a artificial light. So the idea was you would place a candle over here or another light source over here, like a hurricane or something. And that light would be reflected here and then projected onto the lens. Another thing that you have to watch out is that you should never expose this to direct sunlight, because direct sunlight might damage your eyesight. About the microscope, there are two common versions of microscope. One, the more common one is this one. It's called the light microscope or the optical microscope, একই কথা. And the second variety is electron microscope. Now the optical microscope, it can magnify something 1,500 times or 1,500 times. Okay? Whereas an electron microscope can magnify it 500,000 times. Which means if the actual object is 1 millimeter, it will appear to be 500,000 millimeter. Five meter কতটুকু এই রুমে চেতে বলো? Okay, so what's the basic difference between these two? From the name you can understand, light microscope uses light wave in order to focus an object. Whereas electron microscope uses waves of electron to visualize something. Now, I'm not sure if you have studied this in physics, you'll know something like this, that say this is a light wave. Every light wave have their fixed wavelength, right? In order for an object to be visible, the light wave must be shorter than the object. So imagine that this is say a 2-unit wave. If the object is 4 unit, then this light will be able to produce an image of this object. Understood? Yes. However, if this light is 5 unit, whereas the object is 4 unit, this light will not be able to detect this object. Now, what is the average wavelength of the visible light? Let's say 700 nanometer. Which means optical microscope will not be able to pick up objects which are smaller than 700 nanometers. Right? So the figures might not be exactly 700 nanometer, but the concept is there. Which means that these optical microscopes or light microscope has very limited power of magnification, and that's why we can only use it to zoom an object by 1,500 times. Yes. Is there any difference? Both light are the same, right? We light যেটা আছে সেটা আমি দেখতেছি. Okay. So basically the thing is visible spectrum. Light microscope uses the visible spectrum of the electromagnetic spectrum. In contrast, electron microscope, instead of using the light wave, what they use? Electron wave. Now, electron waves are much smaller than the visible light, which means they can pick up objects which are much smaller than 700 nanometer. And because of that, they have a much greater magnification power. Is that clear? So which has better magnification power? Electron microscope. So we will come to electron microscope, but first let's study about light microscope or the optical microscope. In this chart, you can see the advantages and disadvantages of optical microscope. Now, before going to that, let's look at, here are some pictures that were taken by light microscope. প্রথম picture কিসের? How do you know this is a plant cell? Yes, first of all, they are regular, they have a rectangular pattern, and some of them has outline forced vacuoles. So these are very likely to be plant cells. These are some animal cells, and probably they are from the intestine because you can see evidence of microvilli. What is this? These are blood cells, right? So these ones are their red blood cell, and these ones are the white blood cells. And here are some like something like platelets. Any idea what this is? No. No. These are the pathogens of cholera. Basically, they have this coma-shaped structure, and they have a tail, so they can actually wiggle around. Now, one thing you can clearly understand, yes. Yes, these are prokaryotic cells, these are bacteria, which means they are much smaller than all of these cells. Now, one thing that should be clear to you is the very limited magnification power of light microscope. Yes, first of all, we can't see anything inside the cholera cells. We can barely see the tails. If you look at this one, we can only distinguish between the cytoplasm and the nucleus. Same over here. We can't see the Golgi body, we can see endoplasmic reticulum, we can't even see the ribosomes or the mitochondria even. Same over here. So, light microscope produces very low resolution image, right? They have very limited magnification power. So, about the advantage and disadvantage of light microscope, the first disadvantage is the limited power of resolution. It has very limited magnification power. The other thing is that it produces artifacts. Now, what does artifact mean? What is the natural color of the cell? Do they really look blue or purple like this? No. What is the actual color? They're basically transparent, right? So if you have a transparent cell, transparent cytoplasm with a transparent nucleus, internal structure গুলো কি কিছু বুঝে যায় তো? না। এখানে আমরা বুঝতে পারতেছি কিভাবে বুঝি? Because before taking this image, we added some stains or added some color. Because of that color, various different parts of the cell picks up different shades of that color and then they become visible and easily identifiable. This is called staining. We will talk about staining later. But for the time being, just know that in order to take an image under light microscope, we will need to add some stains. And here is the problem. When actual color when we are adding stain, there is always the chance that the stain will also contain some impurities. Right? It might contain some solid particles and dust, some lead particles, cadmium particles, or things like that. If they get into the picture, or if they get into the tissue, they are going to produce a structure called artifacts, and that is going to confuse the observer. For example, if you look at this particular structure, what is this? Is it another nucleus? Is it a bacteria? Is it a part of the cell? We don't know. It could be a lump of lead. It could be a lump of iron that was present in the stain. So these impurities produces what we call artifacts. And so this is another disadvantage of using light microscope. So what are the advantages? Okay, it is relatively light. That means it can be carried in your backpack. So you can be Dora the Explorer and carry around the microscope. It is also very cheap, right? So that's why most of the schools have light microscope. It's like several thousand taka, which is quite affordable, right? And I think the best advantage of having a light microscope is that using a light microscope, you can observe live specimen. For example, if you look at this picture, you can see these are also Vibrio bacteria, and you can see them moving. This one is giving a breakdance, right? So you can observe live cells using light microscope, which means you can see their movement, you can see their lifestyle, how they're invading the cells or how a cell is dividing, you can see all that live. Next comes electron microscope. So this is an electron microscope. So it's almost the height of the scientist, right? You can actually carry this to a battlefield. Rocket launcher শেষ হয়ে গেছে, you can just carry this around and throw it to a tank. It's going to cause some serious damage, right? Okay, so first thing is that it is massive, much bigger than a conventional optical microscope. And it also has some complex mechanism. So it has to have an electron producing source. it needs to produce a beam of electron. So whatever the thing is, we don't have to know the details of this. Basically, the thing is, it is going to produce a wave of electron, which is going to be reflected by a series of lens, refracted by a series of lens, and eventually the waves will be picked out by a detector, and these signals will be synthesized using a computer and eventually produce an image on a screen or a computer monitor. I don't think they're glass lenses. These are probably electric coils. Because here the target is to focus a beam of charged particles. So if you put a glass, it's going to obstruct the electrons, right? So it has to be hollow. Probably this is a lens, a coil of some kind. Anyways, so here you place an ant and a detailed image of that ant will be produced. Now the thing is, again, coming to the advantages and disadvantages, the major and the only advantage of electron microscope is that you can produce very high magnification images with very good resolution power. Everything else is a disadvantage. করতে হয় আরো বেশি করতে হবে first of all it's huge, right? And not just that, in order to operate an electron microscope you need to control the air pressure around you, also the temperature. So buying an electron microscope is not enough. You also have to design a room in order to hold that electron microscope. So definitely you cannot be Dora the Explorer with an electron microscope in your backpack. Next, it's extremely expensive. The specimen has to be treated very elaborately, which leaves more chance for artifacts. এর আগে তো আমাদের শুধু stain add করতে হয়েছিল, and stain ছাড়াও আরো অনেক কিছু add করতে হবে, তাহলে artifacts-এর chance অনেক বেড়ে যাচ্ছে. Next, this time we are using beams of electrons. Electron shows wave particle duality. These are not just wave, these are also particles. So if there is air within this tube, electron গুলো কি হতো? They will get scattered, right? তার মানে এই টিউবের মধ্যে air রাখা যাবে না. The tube has to be in vacuum. তার মানে স্পেসিমেনটা কোথায় আছে? Vacuum. So if the specimen is kept in vacuum, do you think this is live specimen? So using electron microscope, can we observe live specimen? No. So this is another major disadvantage of electron microscope. Okay? Any questions so far? It depends. It depends on many factors. It depends upon your budget. It depends upon the quality of image that you need, and many other things. For example, say you are going to a remote village. এখানে electricity or নাই? ওখানে তুমি কি use করবা? Obviously, optical microscope. So optical microscope is very good for field research. But then optical microscope, one of the greatest disadvantage we have already mentioned is what? Magnification and resolution. And because of this reason, until the 1960s, I think, 1960s or 50s, scientists didn't have any idea about virus. Virus was only discovered after not discovered, observed. Scientists predicted about the existence of virus, but they observed virus for the first time after the discovery of electron microscope. Because viruses are so small, light microscope will not be able to pick them up. So if you're a virologist, if you're researching on virus, then you need to have an electron microscope. Okay, so here are some things about electron microscope. We have already mentioned that they use beam of electrons instead of light because beams of electron have very tiny wavelength. This gives a greater resolution and magnification power. But the disadvantage is that the specimen must be observed in vacuum. That's why they're always dead. Preparation needs a lot of effort and a lot of time, which is also operating the device is very complex. And we can see the image directly, like we can do in optical microscope. The image is projected on a monitor. Now, interesting thing about electron microscope is that the original images generated by electron microscopes are always black and white. Why? Exactly, because we are using electron wave, they are not going to produce color. Later, the computer sometimes can artificially add color to it. So you want to highlight different segments of a cell. তখন তুমি artificially, just like using Photoshop, we artificially add color to distinguish cell organs or other stuff. Now remember these pictures, the plant cell, blood and cholera. So these are the same pictures taken using electron microscope. Those were taken using optical microscope. These are taken by electron microscope. You can see the details of the red blood cell. This is the vibrio coloring. So you can see the comma shape and the tail. You can also see the design on the surface of the bacteria. And this is a plant cell, and you can see the very detailed structure. You can see the nuclear membrane even over here. You can also observe the nuclear pores, the caps, nucleolus. You can see the membrane of the vacuole. You can see the chloroplast, not just chloroplast, the tubes inside the chloroplast as well. This has huge resolution and magnification. Theoretically, you should be able to produce video, but the object is dead, so it's not going to move. So there is no point in making a video. Okay, yes, all of them are dead. The other thing that you need to know about electron microscope is that there are basically two main varieties of electron microscope. The first variety is called transmission electron micrograph, and the other one is called scanning electron micrograph. So how many types of electron microscopes are there? Two. What are there? Transmission electron and scanning electron. So what's the difference between them? Transmission electron micrograph data, it produces a 2D image of the object. However, this has very great magnification power. In contrast, scanning electron micrograph, they have lower magnification, but they can produce a 3D image. I'm going to show you examples, then you will understand. But you have to keep this in mind, that transmission electron micrograph, it produces what kind of image? 2D. And what is the power of magnification? Higher. In contrast, scanning electron produces 3D image, but has lower magnification power. And like we mentioned, sometimes false colors are added to highlight structures. So these pictures shows the difference between the two types. This one, you can see, is it 3D or 2D? 3D. This is 3D and this is 2D. So you can see the difference, right? Here again, this one, 3D, this one, 2D. And you can also see that this has lower magnification, but this has much greater magnification. Same goes with this one. These are, I think, bacteria, E. coli bacteria. This is a 3D image, as you can see, and this is a 2D image. And these are mitochondria. Again, this is a transmission electron micrograph. You can see the 2D structure. You can see them in great details, huge magnification. And this is the same mitochondria, but viewed using the scanning electron microscope. Yes, these colors has been artificially added. These are the original images, and later color can be added. Yes, you can actually produce sections of images. In this topic, you need to have some mathematical skills. Sometimes you will be given maths in your exam. The first thing you need to know is how many lenses does a conventional optical microscope have? 2. Objective lens and eyepiece lens. So imagine that the objective lens has 10 times magnification and the eyepiece lens has 10 times magnification. So the total magnification will be a product of these two. That is 10 into 10, which is 100. Say for example, if this was 20, what would be the total magnification? 200. The second, yes. Yes. Second skill that you need to acquire is this formula. I think you have learned this from physics. There is a formula in physics. Magnification is equal to the actual size, image size by actual size. Right? Did you study this? Magnification equals image size by object size. Now, if you rearrange this equation, you're going to get actual size is equal to image size by magnification. So you need to use this formula, remember this formula, and rearrange it. Sometimes they might ask you to find the magnification, sometimes they might ask you to calculate the image size, or they may even ask you to find out the actual size of the object under the lens. Here is an example. If the diameter of a cell measured under light microscope at a magnification of 400 times is 1 centimeter or 10 millimeter. So here are the key information. This is we know the magnification. We also know the image size. We have to find out what? Actual size. So we know the formula. Actual size is what? Image size by magnification. So that gives us 10 millimeter divided by 400, and that leaves us with a object size, that leaves us with an actual size of 0.025 millimeter. What is important in this type of maths is you have to look at the unit. Yes? Okay. Imagine this is a egg of an insect. What is the actual size of this? Say 5 millimeter. Now when you are going to observe this under microscope, this will look something like this. And we are going to see it soon. Inside the microscope, there are some built-in scales. So while you are observing the object, you can also measure the size using that scale. So imagine the ekhane so 50. So actually, how much is it? And the image produced seemed like it was 50 millimeter. Then you can do the calculations to find out what was the magnification of the lens. Or if you know the magnification of the lens, if you know the new size, that is the image size, you can use the magnification and this one to calculate the actual size of the object. They will give you two parameters, at least two parameters, and you have to work out the third parameter. Sometimes image size jodi na deya thake, you have to use your own scale to figure out. Try to use the centimeter scale, convert it into millimeter. Okay, and we are going to find these questions as we will be solving the question papers. তখন আমরা আরো ডিটেইলসে ডিসকাস করতে পারবো। We just mentioned this, that some microscope has some built-in scales. These scales are called graticule. Now there is an experiment in the unit 3. We will discuss this in more depth when we'll go into the unit 3 section. For the time being, just know that some eyepiece has these built-in scales. These are called the graticules. And using graticules and some mathematical formula, we can eventually use this data to calculate the size of the object that we are viewing. Understood? Yes. And we also need to know the difference between the word magnification and resolution. Magnification means যে একটা object কতটা বড় লাগতেছে. So if the original size of the object is say 1 millimeter, but it appears to be 2 millimeter, it means it's twice as big. It appears twice as big. That means it has been magnified 2 times. And what does the word resolution mean? Clarity, yes. Basically, it's the ability to differentiate 2 points. For example, if I draw 2 dots, there is a gap in between. Can you see that? Yes. There are actually 2 points separated by a very tiny gap. You can see it, say, from 5 meter distance, but if you had watched it from 10 meter distance, do you think you would be able to see the 2 points, or would it appear like a single point? So this is the concept of resolution. High resolution camera are better at distinguishing individual points. So here are some examples. This one was taken by a low resolution camera, producing a fuzzy image or a very blurry image. and this one was taken using a high resolution camera. You can see the individual protein molecules. Same over here. This is again a cell, and this is the nucleus. Low resolution picture seems that the nucleus is made of a homogeneous material, but when you take a high quality image, high resolution image, you can see the individual membranes within the nucleus. Focusing is a part of resolution, right? And here again, I think this dot represents individual atoms. This is a very high resolution picture where you can separately count the atoms, and this is the same picture but taken using a low resolution camera. এখানে কিন্তু individual atomগুলো বোঝা যাচ্ছে না. So that's the difference between magnification and resolution. And about staining, we talked about this at the beginning of the lecture, that in order to see the internal structures more clearly, we will have to add some colors. Colors গুলোকে আমরা stain বলি. In order to demonstrate how important staining is, you can use these pictures. These and these are the same pictures. This one has been taken without stain, and this one was taken with the stains. You see, before stains were added, you couldn't actually identify the white blood cell, could you? Why? Because the nucleus was not visible. Now after the stain is added, you can clearly see the outline of the nucleus. You can also clearly see the outline of the cell membrane. You can also see some of the internal structures. So you can understand how important this staining is.
Exactly, science কিভাবে বুঝবে এই জিনিসটার কালার এটাই স্টেইনস বা মানে স্টেইন না করে তো অন্য কালার আছে যখন নাকি স্টেইন
original color to transparent, right? Yes, this is original color without stain. বুঝিনি. It is because of this is because of the material that the structure is made out of and the type of protein they contain. If you want to know about the details, each of these stains, they have certain acidic or basic property. So some part of the cell will take up the acidic stain, the other parts will take up the basic stain, giving the cytoplasm a pinkish color, whereas the nucleus a bluish color. Yes. No, it's not just because of the nucleic acid, there are many things. But the thing is, due to composition of each of the structures within the cells, they have affinity towards different type of stains, and that's why they take different colors. So here is another example. This part was taken without stain, and these pictures were taken after adding the stains. So you can see the differences. And actually we have run out of time. There was another maths. It's also in the book. Page number 150. So you can find this at page 153. Okay, so any questions? Then that's it for today.